Let's dive right into one of the biggest paradoxes in modern agriculture. I'm talking about our incredibly complex relationship with nitrogen. We're going to explore how cutting-edge science is trying to fix a cycle we broke, using a process that's both a hero and kind of a villain in our global food story. So get this, the Haber-Bosch process, which is the industrial way we make synthetic nitrogen fertilizer, is credited with feeding this many people every single year. I mean, this is the chemical reaction that literally pulled humanity back from the brink of famine a century ago and kicked off a massive population boom.
But of course, this modern miracle has a pretty staggering cost. The process is so energy intensive that it eats up between 1 and 2% of the entire world's energy supply each year, and most of that comes from fossil fuels. It's also responsible for about 1.2% of all global carbon emissions. And this quote just captures the dilemma perfectly, right? We have this process that underpins the diet of almost half the world's population, but at the same time, it's pushing our planet's boundaries right to the limit.
So in this explainer, we'll first break down this whole nitrogen dilemma and check out some of the smart solutions that are already being used. Then we'll get into the really cool stuff, how CRISPR gene editing could completely change how plants feed themselves. And finally, we'll weigh these strategies against each other and see what the road ahead looks like.
Okay, so let's start with the real core of the problem. It's not just about the energy it takes to make the fertilizer. It's about how unbelievably inefficient we are at using it once it's actually on the farm.
The sheer amount of waste here is just staggering. On average, our crops only manage to absorb about half of the expensive, energy-intensive nitrogen fertilizer we put down. And the other half? Well, it doesn't just vanish. It runs off, and that creates this whole cascade of environmental disasters.
So this excess nitrogen leaches into our rivers and lakes, causing what's called eutrophication. These huge algal blooms that just suck all the oxygen out of the water, creating these vast aquatic dead zones. And in the soil, it can cause the release of nitrous oxide, which is a greenhouse gas about 300 times more potent than CO2. Oh, and it makes the soil more acidic, which isn't great for long-term farming.
So, faced with this massive challenge, scientists and farmers have started developing much smarter strategies. They rely less on just brute force chemistry and a lot more on ecological intelligence. Things like splitting up fertilizer applications is a decent first step, sure. But the real game changers are practices like no-till farming and planting cover crops. They go way further by building up healthy soil that holds on to nutrients all by itself. This healthy soil is really the foundation for everything else we're about to talk about.
And the most fascinating part is that nature actually solved this nitrogen problem billions of years ago. It's called biological nitrogen fixation. There are these specialized microbes out there that can do something incredible. They take nitrogen gas straight from the atmosphere, which is like 78% of the air we breathe, and fix it into ammonia, which is basically a natural fertilizer for plants. And just harnessing this natural process is unbelievably powerful. If you take a legume field, think soybeans or chickpeas, and you just add these helpful microbes, you can boost yields by up to 25%. That's a huge win, and it comes from just working with nature, not against it.
Okay, so working with microbes is a huge step forward. But what if we could build these capabilities right into the plants themselves? What if we could actually edit a plant's own operating system to be more efficient? This, right here, is where the real revolution begins, and it's all thanks to a technology called CRISPR.
This is the big question that scientists are now able to answer with CRISPR. Instead of just dumping more nitrogen into the system, we can re-engineer the plant to make much better use of the nitrogen that's already there.
So with CRISPR, we can go down two main paths here. Think of it like a light switch. The first strategy uses a tool called CRISPR activation, or CRISPR-A. This is like turning a dimmer switch up. Scientists find the genes that are positive regulators, the ones that help the plant absorb nitrogen, and they crank up their expression. The second strategy uses CRISPR interference, or CRISPR-I, and that's like turning a switch completely off. We find the negative regulators, the genes that are basically putting the brakes on nitrogen uptake, and we just silence them.
And here's how that actually works. First, scientists pinpoint those key regulatory genes. Second, they design something called a guide RNA, which acts like a GPS, taking the CRISPR tool to that exact spot in the plant's DNA. Third, they use the right tool for the job, either activation to turn a gene up or interference to turn it down. And finally, they grow a whole new plant from these edited cells. And voila, you have a crop that is naturally, intrinsically better at using nitrogen.
But the ultimate ambition, the absolute holy grail of this research, is even more mind-blowing. The idea is to take that whole cluster of genes, the NIF genes, that let those bacteria fix nitrogen, and copy-paste them directly into the genomes of our most important crops, like wheat, rice, and corn. Just imagine a corn plant that can make its own fertilizer. That's the end goal.
So we have these three powerful toolkits, right? We've got farming practices, microbial helpers, and direct genetic editing. How do they all stack up against each other? And more importantly, how do they fit together?
When we put them side by side, you can see there are clear trade-offs. Agronomic practices are great because they're scalable right now, but they can be a bit imprecise. Microbial inoculants give a fantastic biological boost, but they really depend on the soil being just right. And the genetic strategies, they offer incredible precision, but getting them developed and out into the world is expensive and complex. It really suggests the future isn't about picking one, but about integrating all three.
And this is where it all comes together in this beautiful virtuous cycle. Sustainable farming practices like no-till build healthier soils. That healthier soil creates a much better home for beneficial microbes, like those nitrogen-fixing bacteria and mycorrhizal fungi. And in turn, that healthy, microbially rich soil allows the genetically enhanced root systems of these CRISPR-edited crops to really, truly thrive. Each part just makes the others stronger. Of course, having amazing science in a lab is one thing. But getting it into the hands of millions of farmers all over the world, that is a completely different challenge.
Yeah, beyond the purely technical stuff, the biggest hurdles for these advanced genetic solutions are, 1, navigating the super complex country-by-country maze of government regulations, and 2, just as important, earning public trust and acceptance for these new technologies.
This table gives us a fantastic roadmap of what's possible right now versus what's still on the horizon. See, microbial inoculants are already on the market and being used today. Engineering new symbiotic relationships is showing some early success in the lab, but that really ambitious goal of transferring nitrogen-fixing genes directly, that's still in the very early experimental stages.
So what's the top priority for today? Well, the most practical path forward is to really focus on what we know works, combining those commercially available microbial solutions with proven, sustainable farming methods. This can give us real benefits right now, while we continue to develop the next generation of truly revolutionary genetic tools.
And all of this leaves us with this one final critical question. The first green revolution, the one powered by the Haber-Bosch process, fed billions of people, but it came with some pretty serious hidden environmental costs. Now, as we stand on the edge of a second one, powered by genomics and biology, the real challenge is to use this incredible technology wisely, to make sure we build a food system that's not just productive, but truly sustainable for the future.